The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome. In today's lesson, we will talk about individual differences. We'll start with historic surveys of individual differences across the world. The first worldwide human genetic diversity survey is the Human Genome Diversity Project, or HGDP, that was started by Stanford University Morrison Institute and a collaboration of scientists around the world. This was the result of many years of work of the famous scientist Luigi Cavalli-Sforza, one of the most cited scientists in the world, who has published extensively on the use of genetics to understand human migration and evolution. On the map, you see sites indicated where the HGDP samples have been collected. The next project, the International Hapmap project, was organized to develop a haplotype map, the map of haplotypes in different populations of the human genome, and to describe the common patterns of human genetic variation. The Hapmap originally included 4 populations, but then in the second phase, several more populations were added. At this time, there was a big interest in developing commercial genotyping platforms, and the problem was that sometimes platforms that were developed for one population didn't work very well for another. This is called the ascertainment bias. Ascertainment bias in genetics is a systematic distortion in measuring the true frequency of genetic markers due to the way that the data was collected. As an example of addressing this problem, the Affymetrix Axiom human origin assay was designed. This was designed using International Hapmap. This array includes 630,000 SNPs ascertained by resequencing in 13 populations of HGDP and Hapmap. The next big project took it a step further. The International Hapmap project was only focusing on certain population in the world, but 1000 Genome Project was trying to capture far more detailed catalog of human genetic variation. The Hapmap was followed by 1000 Genome Project somewhere around 2008, specifically in January 2008 was an inaugural meeting. The goal of 1000 Genome Project was to find the most genetic variants with frequencies of at least 1%. These are so-called common genetic variants in 26 worldwide populations. These are shown on a map on the left. The project took advantage of development in next-generation sequencing technology, so while in Hapmap they used genotyping, 1000 Genome Projects used NGS because of its sharply reduced cost. So, what have we learned from surveys of genomic variation around the world? We learned that world populations differ in allele frequencies. Alleles occur at different frequencies in different human populations due to a combination of evolutionary forces. Populations that are geographically and ancestrally remote tend to differ more. Studies have demonstrated that thousands of individual SNPs distributed through the genome have very large differences in allele frequencies between one or more continental populations, such as between Europeans, Native Americans, Asians, and Africans. There are several examples of markers that differ significantly between continental populations. One of the famous markers is called Duffy null allele. This one is denoted by FYO, and it has a frequency of almost 100% in sub-Saharan Africans, but occurs very infrequently in populations outside this region. A person having this allele is thus more likely to have sub-Saharan African ancestors. Each segment of the chromosome can therefore be assigned to ancestral population based on the number of alleles such as Duffy null allele on the chromosome. And if you examine a suite of these markers that are evenly spaced through the genome, it is a cost-effective way to discover genes underlying complex diseases in a technique called admixture mapping, or sometimes referred to mapping by admixture linkage disequilibrium. We'll address this elsewhere, but today we'll look at ancestry informative markers such as Duffy allele. So in general, these ancestry informative markers are single nucleotide polymorphisms that exhibit substantial different frequencies between different populations. Collections of AIMs have been developed that estimate the proportional contribution of each geographic origin of ancestors in each individual. AIMs are selected by filtering SNPs for the most differentiating frequencies in the population and then distributed across the chromosome. In this way, individual admixtures can be calculated from the fractions of population-specific ancestry informative markers, which is referred to as global ancestry proportion. Ancestry estimates can be extrapolated on geographic map to understand population history. Here is the map from our paper called "The History Shaped the Geographic Distribution of Genomic Admixture on the Island of Puerto Rico." As you can see from the map, there was significant geographic variation of ancestry across the island. In particular, African ancestry demonstrated a decreasing east to west gradient and was partially explained by historical factors linked to the colonial sugar plantation system. Admixture estimates can also be calculated for each chromosomal position based on the AIM. In a technique called chromosomal painting, alleles representing ancestral haplotypes are assigned ancestry based on the chromosomal location. Another interesting consequence of recombination is that fragment size can indicate the age of admixture. Because admixture is constantly happening, the older is the admixture, the smaller are the resulting fragments. If you find fragments that are very short, that indicates that admixture with that continental population has entered into your genome long ago, while if the fragments are large, the age of them is recent. Here's an example of an individual whose chromosomes have been analyzed by local ancestry with AIMs. This specific individual has appeared to have equal Eastern European and Balkan admixture. The longer fragments indicate a more recent admixture, while short fragments indicate the older one. I prepared some review questions for this lecture. Can you describe methods of the source of Human Genome Diversity Panel? What was the goal of the International Hapmap? What were the goals of 1000 Genome Projects? What is an AIM? Contrast between global and local admixture. How can you estimate the timing and the source of the admixture? This concludes today's lesson, Individual Differences. Next, we will be looking at the topic of population differences.